Our next speaker, Professor Atsushi Miyawaki, will be more on the protein problems, fluorescent problems, which is closely related to the microscopy imaging, and it's really another important aspect. So Professor Miyawaki is the Deputy Director of the Recon Brain Science Institute in Japan. So after his PhD with the Osaka University, he pursued postdoctoral studies on the Nobel laureate Roger Yang at University of California, San Diego. So he is a leading expert on these fluorescent proteins, and his group is currently interested in the development of new bioimaging technology using the fluorescent proteins. So today, Professor Miyawaki will talk about the new fluorescent and the new perspective in the bioscience. Okay, so let's welcome Professor Miyawaki.
Okay. So I'm Atsushi Miyawaki from Japan. So, thank you. All right. So first, I'd like to thank the organizers for the very kind invitation. I enjoy myself very much so far. Okay. So, you know, many biological events are well organized, sometimes very ingeniously controlled in space and time. The cruising inside cells. So that's the title of my talk. Okay, all right. So so many events are controlled in space and time, and my laboratory is engaged in the development of imaging technologies, so using fluorescent proteins, and those technologies allow us to get insight into spatial-temporal dynamics of many important biological functions, and within the body, a cell, a tiny domain. All right, so I'd like to start my talk with this movie. Something, moving. So do you have any ideas what they are? Guess what? Shout? Jellyfish? No. So they are osteoclast, so which are responsible for bone resorption. Okay, and the osteoclast has multiple nuclei. And also it has an actin ring in the periphery. Okay. So we fused a mutant of E. coli GFP, so GFP from E. coli Victoria, so to an actin protein, so in order to label this cytoskeleton structure. And after gene transfer, so we performed long-term, time-lapse imaging, so to observe the behavior, so the differentiating osteoclast. So these cells become multinucleate cells through cell-cell fusion. Okay? And upon fusion, so the actin ring does collapse, so transiently. So the imaging, fluorescence imaging experiment, so it's very fun to do, so very exciting. So now let's back upon the ecology briefly. Okay? So as you know, Dr. Osamu Shimomura in 1962, so he discovered purified GFP from the light-emitting organ of jellyfish, and almost 30 years later, so in 92, so its cDNA was cloned. Okay. And Dr. Shimomura collected a huge number of jellyfish at the Friday Harbor Laboratory in Washington State, so near Seattle. Okay. And so the bottle which I held contained GFP purified by him and from 200,000 jellyfish. So very precious protein sample. Okay. And amazingly, the protein can be spontaneously fluorescent. It doesn't require any cofactors or small chemical compounds to become fluorescent or to generate its chromophore. Therefore, so the heterologous expression of GFP was achieved by Marty Sharpie in 1994. Okay. So the chromophore, so it's a part of the molecule which can interact with visible light. Okay. And so the wild type E. coli GFP is composed of just 238 amino acids. Okay. And its chromophore is formed from these 3 amino acids. Okay. Serine, tyrosine, glycine, SYG. So that's the tripeptide for chromophore formation. Okay. And while the peptide chain falls into a native conformation, so beta bottle, so the 3 reactions occur spontaneously. Okay. So cyclization and dehydration and oxidation reactions, so to make a pi conjugated structure. Okay. So now the pi electron is delocalized over the structure. So this is the GFP's chromophore, so which absorbs blue light and emits green light. Okay. And the chromophore is buried inside the beta bottle structure, so which consists of 11-stranded beta bottle with 1 alpha helix inside. And the chromophore is placed on the helix, and with some packing, so the chromophore can take a rigid planar structure. So that explains why GFP has high quantum yield, fluorescence quantum yield. Okay. And then also mutagenesis studies have produced several very useful GFP mutants of different colors. So done by Dr. Roger Chen at UC San Diego, my previous advisor. So I was in his laboratory from 95 to 98, and I enjoy myself very much. So he gave me many pieces of advice, intellectual advice, and he's very special for me. Okay. And as you know, so the 3 people won the Nobel Prize in chemistry in 2008. Okay. All right, so going back to 1984, so a long time ago, when I was a student, so like the young people in the audience, so I was very much interested in the regulatory mechanism of transcription in prokaryote. So I just wanted to demonstrate the direct interaction between these 2 proteins, RNA polymerase and some transactivated protein. And for that, so I took interest in FRET, so which was explained by Shimon, so this morning. And FRET is a transfer of the excited state energy from a donor to an acceptor molecule. And when I was in San Diego, I utilized CFP and YFP as the donor acceptor of FRET, so to make an indicator for the calcium ion, the chameleon. Okay? And so after transfection, chameleon into cells, culture cells, I observed calcium oscillations. And after stimulation with histamine. So I believe oscillatory changes in calcium concentration is now taking place in some cell types of the body. Okay. And it's so interesting to know how extracellular stimuli are encoded in such temporal profile and how cells decode the information for various downstream events, cellular events. Okay. And when we increased spatial temporal resolutions, so now what we were able to observe propagation, so calcium propagation within individual cells. Okay. And zebrafish. Okay, already some speakers mentioned or touched upon the zebrafish. So its genetics is well established, so we can easily make a transgenic line, so producing chameleon. And so we studied calcium dynamics, so very early embryogenesis of fish, and after fertilization, so the calcium dynamics is mostly linked to cell division. Okay. And the calcium transient precede the cell cleavage, maybe in order to activate calcium dependent many machineries in advance. And personally, I liked the calcium fluctuations, which are not related to cell division like this. Okay. And also, when we use 2-photon excitation microscopy or nonlinear optics, so we could extract the calcium signals very deep inside the animal. Well, much later stage, in the segmentation period, or 25 somite stage, so you may be able to identify node code and the neural tube and fluoroporate, and we observed calcium oscillations in almost specifically in the fluoroporate for a long time, so over 4 hours. And each calcium transient lasts about 20 seconds. So, but we haven't the slightest idea. So, what is the significance or so the mechanism for the spatial temporal pattern? So, it's still riddle in our mind. Okay. But so far, both CFP and YFP, so coming from Equator Victoria, okay, and not only the jellyfish, but some other cnidarian animals, so can produce fluorescent proteins. And now we researchers can use a huge number of fluorescent proteins, and so Equator GFPs, so from Equator Victoria, and GFP-like proteins from other animals. So mainly coral animals, corals. Okay, and the most famous one is DS red RFP. Okay, and it's in DS red. So not SYG, but QYG, glutamine, tyrosine, glycine. So these 3 amino acids, so the tripeptide for chromophore formation. Okay. And again, so through the 3 reactions, cyclization, dehydration, oxidation reactions, a green chromophore is formed. And then an additional oxidation reaction occurs here. So to make or to extend so the pi conjugate structure, so up to here. Okay. So that the more extended, so the redder or the longer wavelength. Okay. And since 1999, so we occasionally went to Okinawa Island, okay, south part of Japan. So Okinawa has a very rich biodiversity. Okay. And we dive into the water. And I'm very bad at diving. Okay. And we collected so many large number of coral animals. And from those animals, we have cloned new fluorescent proteins or new chromoproteins. Okay. And fluorescence has many interesting or important properties. Okay. So the brightness fluctuation or spectrum or energy transfer, lifetime of the excited state, anisotropy, photoconversion, chromism, bridging and quenching, etc. Okay. So we can make the best use of these properties to create new imaging modalities. Okay. So let's start with photoconversion. Okay. So about 10 years ago, so we went to a pet shop in the downtown of Tokyo and to meet. It's a very colorful stony coral, Trachyphilia. Okay. And we bought it, so it was $20. Okay. And at first glance, so I thought, so we should be able to get multiple proteins, so more than 1 fluorescent proteins, so just because it was so colorful. But I was wrong. Okay. So technician in my lab, so Ryoko Ando cloned so some fluorescent proteins, but all of them were green. Okay. But we had 2 lacks. So first, so her lab bench is closest to the south-facing windows. Okay. And the second, she comes to lab very late. Okay. She shows up at 5 or 6 p.m. every day. Okay. So the one night, so she happened to leave the protein sample on her lab bench before going home, and the next day, so the tube had been exposed to sunlight substantially, and then she came and found that, so the protein had turned red. Okay. So the late bird gets the worm. Okay. All right. So this is photoconversion, a change in spectrum induced by specific light, and the change is irreversible. Okay. And our protein changes color from green to red upon irradiation of violet light or ultraviolet light, UV or V. Okay. So we named the protein Kaede maple leaf in Japanese. Okay. All right. So now, so these 3 neurons, culture neurons, expressed Kaede protein, and initially green everywhere. So it was so difficult to delineate individual neurons because they extend long processes that become entangled. Okay. But using a laser diode 405, so 405 nanometer violet laser diode, So these two somata were illuminated for 0.5 and 0.25 seconds, and then we could enjoy the autumn colors on the petri dish. Okay. And it's so easy to make a transgenic zebrafish expressing kaede, and we can optically highlight any cells, and then we can follow their fate. Well, I mean, in other words, cell lineage analysis is very easy to do. Okay? And we clarified the molecular mechanisms of the green to red photoconversion. Okay? And now, kaede has HYG. Okay? So HYG, histidine, tyrosine, glycine. And histidine has imidazole ring as a side chain. Okay? So this is very important. And again, through those three reactions, okay, so cyclization, dehydration, oxidation reaction, so the green chromophore is formed, and then the UV irradiation brings about the change in charge distribution like this, and the cleavage of a peptide chain. Okay, so the light-induced cleavage of the main chain. Okay. So this is the beta elimination reaction. Okay. And the leaving group is carboxamide. And then now, introduction of new double bond here, then connect the imidazole ring of the histidine to the original chromophore. Okay, so now the pi conjugate structure is expanded to make a red chromophore. Okay. So this photo-induced cleavage of peptide chain is so interesting to us. Okay? And in biochemistry community, okay, not chemical, but the biochemistry community. So when it comes to the cleavage of the peptide chain, so it occurs at the peptide bond here, okay? So, and through hydrolysis, okay, at the peptide bond and through hydrolysis. But in the kaede protein, so the light-induced cleavage occurs not here, but here, so between C alpha and immunonitrogen. So this covalent bond is so stable, so the cleavage here should require a huge amount of activation energy. Okay. And as you can imagine, so the whole kaede protein can act as a catalyst to reduce the activation energy. And so the catalysis is specifically triggered by the absorption of UV light, by the chromophore. So this is a very interesting molecular machinery. So we are always trying to understand how electrons feel over the chromophore within the molecule. Okay. And photochromism. So photochromism is now reversible. So it's defined as a photo-induced reversible transformation of a molecule between two forms. Okay. So after our paper on kaede came out, so Ryoko came up to my office with a new coral, very shabby coral, dirty coral. Okay. And she said she would be interested in cloning something from this coral. And I was negative because it was dirty and compared to Trachophilia. Okay. So, but she did that. And then she cloned very bright green emitting fluorescent protein. Okay. And after that, well, after relentless mutagenesis, so she made it photochromic. Okay. So now we can convert the protein between the dark and bright state, so using two different two colors light. Okay. So reversible photo switching. Okay. So we named the protein Dronpa. So Dron means disappearance in the ninja world, and the Pa stands for photoactivation. And we made the Dronpa protein bacteria and spread it on a kappa strip to make a rewritable molecular memory system. So Eric showed this kind of the slides kindly in previously. Okay. So we had two lessons. One, don't judge a coral scientifically by its appearance. And the lesson two, even if a wild type, so protein is dim or boring, don't give up or don't worry about it. So we can make it, it's possible, so highly possible, we can make it bright beautiful or interesting through molecular evolution, so in test tubes. Or in other words, so in vitro mutagenesis is very becoming powerful. Okay? So that's my point. And so due to the time, so I skip this. Some applications of drop protein to biological questions. Okay. All right, and then I'd like to talk about something within the whole brain, some biological functions in the whole brain, and I'd like to tell you about cell cycle. And as you know, so this is the cell cycle of eukaryotic cells. G1 is due to an M. So in S phase, so DNA synthesized. Okay. And we wanted to visualize the transition from G1 to S, because this transition is very important. It's involved. So it involves very important decision to initiate a new round of DNA synthesis. So we were interested in the transition from G1 to S. And Asako Sawano in my laboratory is the leading person. Okay. And to make an indicator, we used the ubiquitin oscillator. Okay. So cell cycle dependent proteolysis or cell cycle dependent ubiquitination. So the basic function of ubiquitin, or so the component of ubiquitin, so the degradation pathways, so well studied in 1980s, and the Nobel Prize for that was awarded. So for that, so in 2004, I remember. Okay, and we used so the CD21 and germinin. So protein levels of these two proteins oscillate reciprocally during the cell cycle, okay, just due to the protein degradation. Okay. And then we made a cell cycle probe which can label individual nuclei in G1 phase red and those in SG2M phases green. Okay. And we named the proteins of FUJ, fluorescent ubiquitination-based cell cycle indicator. And so Dr. Sharpless, so this morning, touched upon HeLa cells already. It's coming from malignant tumor. Okay? And when we use just a normal DIC method, so microscopy, we can follow their cell division. But with FUJ system, so we can monitor cell cycle progression in real time at a single cell level. And then, in contrast to HeLa cells, now NME cells come from benign tumor. Okay? So therefore, the stable transformants show very nice contact inhibition. Okay? So upon reaching the confluent monolayer, all of the cells became red and stopped their proliferation. Okay? G1 arrest. Okay? And also, we could distinguish between the benign and the malignant features of the tumor cells. So after transplanted or after injection into a mammary pad of nude mice, so so HeLa cells continue always growing. So always, the color of the cell mass was yellow, mixture of the green and red. But benign cells, so NME cells, so stopped their proliferation. But sometime later, after the inoculation, so the nuclei became red. Okay, so we can easily distinguish between benign and the malignant. So just the color, so the cell mass. Okay. And also, so tumor vessels are visualized by loading the animal with the angiosense specific dye. And we can easily, see cell cycle profile. So in the tumor cells. Okay. Well, we believe that this system can provide us with reliable pharmacodynamic readout of growth, invasion, and metastasis of tumors in the whole animal. Okay. And we made a transgenic mouse which expressed FUJ ubiquitously or constitutively. And this is the coronal section of the embryo, E13. Okay? And every cell nucleus shows either green or red fluorescence depending on the cell cycle phase. And for instance, in the brain, so the neural stem cells in the ventricular zone had green or red nuclei. But the post-mitotic neurons had only red nuclei in the cortical plate. So in short, so green signals indicate cell proliferation, and red differentiation. Okay. So for instance, in the eyes, it's in the retina, so we can appreciate centrifugal differentiation of retinal ganglion cells quite easily. Okay. Of course, initially green signals were overwhelming, but with time, green to red ratio decreases. And after birth, red signals become predominant. But even in adult tissue, we can find very few green nuclei. Okay. And the cells with those green nuclei correspond to tissue stem cells. Okay. So in fact, we identify very few green nuclei in the dentated gyrus of the hippocampus, adult mouse. So they are neural stem cells, okay, stained for nestin protein. Okay. And in order to look at cell cycle profile in the whole animal, so we again turn to zebrafish. And to make a long story short, we made a zebrafish-specific food system and transgenic fish line again, and in a segmentation period. So this is side view and head and tail. And the green to red ratio decreases with time, but some organs like retina and the brains were very green. Okay? And the top view, and when we zoom in on the retina, okay, so in its new epithelium, so we can look at the interkinetic nuclear migration. Okay, in other words, green nuclei moved from basal to apical, okay, but red nuclei moved in the opposite direction, from apical to basal. This is the interkinetic nuclear migration. And also in the differentiating node code, we saw cell cycle transition which troubled, okay, from the head to tail. And also we used the EDU, which Dr. Sharpless introduced to us this morning. So this is based on click chemistry. And we successfully visualized the arrangement of notochordal cells in the order of the cell cycle phase G1S and G2 from posterior to anterior. So let me stress again, zebrafish is a very good experimental animal because it is transparent. Okay. So I wish mammalian animals were transparent like fish, okay. But it's turbid. Okay. So then finally, I'd like to tell you our recent technology, scale technique, which enables large-scale and subcellular resolution and three-dimensional reconstruction of biological structures. Okay. So Hiroshi Hama is the central person of the project. So the three-dimensional imaging of large specimens should require sectioning, okay, to improve ZX resolution or access to deeper structures. And we perform optical sectioning, not mechanical sectioning. But the performance of optical sectioning is mostly limited by the scattering light inside tissue. Okay. So the light penetration inside tissue is prevented by two events, absorption and scattering. Okay. So, but in our body, the light scattering contributes to turbidity more than light absorption. Okay. So scattering or Rayleigh scattering, it's inversely proportional to the fourth power, so the wavelength, and it is responsible for the blue color of the sky during the day, and also the red colors during the sunrise and sunset. Okay. And so due to the scattering, very significant scattering, so there is a limitation in the imaging depth. Okay. So we have developed some new techniques or tissue clearing reagent, based on urea, and among them is scale A2. So this is a prototype, and the formula is quite simple. 4 molar urea, 10% glycerol, and 0.1% Triton X. That's it, and based on urea. Okay. And so the aqueous solution can optically clear the tissue greatly after fixation with 4% PFA. So the 2 mouse embryos were fixed And then, so this one was incubated in scale A2 so for 10 days. And please pay attention to the liver or retina, bile acid or retina. So scale A2 does not affect so light absorption at all. So it only eliminates the light scattering. Okay? And the clear brain, so it can be traversed by the light from the laser beam in the dark. So let's visualize neuronal circuit. And I show you some data of YFP-H mouse line. In its brain, YFP is expressed in the subpopulation of neurons. Okay. And here is the whole brain. So cannot see very well, but whole brain here, fixed brain. And we put just objective lens, okay, to visualize, to look at so inside so the brain. And here we visualized YFP expressing neurons down to a depth of 2 millimeters, okay, from the brain surface. Okay. Just because so the objective lens had the working distance of 2 millimeters. Okay. And the spatial resolution was good enough to discern individual dendritic spines at layer 5. Okay. So scale technique allows us to see both the trees and the forest. Okay. And this is a hippocampus in rodent. So hippocampus in the rodent plays an important role in the navigation and the spatial memory. Okay. But its anatomy is very complex. But easily we can, we can appreciate the complex hippocampal formation from many different angles. Okay. And now, so within the transplant brain, so the imaging depth limit is physically determined by the working distances of currently available objective lenses. Okay. So we asked some manufacturers to make a customized objectives with much longer working distance. So one company, so Olympus company provided us new objective with the working WD of 8 millimeter, okay, not 2 millimeter. Now, so we are able to, make a 3D reconstruction that extended from the brain surface to the bottom of thalamus, so through hippocampus. So let's dive deep into the brain. So now cerebral cortex, so soon passing by the white matter, and then see one region of the hippocampus, and then there are neurite, so also dentate gyrus, neurogenesis locus, and then we entered the thalamus. So I'm sorry, so the contrast of the slide displays somewhat low, but we can easily see very fine structures. So I wish I could show you on that display. Okay. And also when we apply scale techniques to food transgenic mice, so we can visualize, we can enjoy the comprehensive perspective, so the cell proliferation versus cell differentiation, and many places and times. Okay. So let me stress again, emphasize again, so many biological events, well ingeniously organized in space and time, and the fluorescence imaging technologies allow us to enjoy that. Okay. And finally, so those technologies, or development technologies, will promote the multidisciplinary, research, implicating the neuroscience, physiology, optics, and the cell biology, developmental biology, chemistry, molecular biology, biochemistry, mathematics, and etc. So for young people, be ambitious and be multidisciplinary from now on. Okay. So I end my talk by thanking all the people, all the members of my laboratory and the collaborators. Okay. Thank you very much.